Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing Central University Common Entrance Test question paper CUET MSc Entrance question paper conducted in the year 2023. This was conducted in 25th June 2023. I have solved the biochemistry portion with explanations. If you are new to my channel, please do like, share and subscribe. I have also uploaded still previous year's question papers of CUET entrance exam. You can refer to my playlist for the same. Coming to the first question. Which of the following statement is true about nucleoside? Now nucleoside, they don't have a phosphate in it. Only nitrogenous base and sugar is present. So the options, that is 2, 3 and 4, can be omitted because it says they have a phosphoryl group attached. So that is not correct. Now if you see, this is the base. It is having an H here. This is the sugar. It is having an OH here. This is the C1 carbon of the sugar, C1 dash carbon of the sugar. This is N ion of this base. So upon removal of the water molecule, you will get N-beta glycosyl bond between sugar and the nitrogenous base. So that is what a nucleoside is. So nitrogenous base attached to the OH of C1 of the pentose sugar. That is the answer about nucleoside. Then which of the following is an amino acid? So tryptophan is an amino acid, proline is an amino acid, lysine is an amino acid, and phenylalanine is also an amino acid. Then which of the following antibodies can cross the placenta? It is IgG which can cross the placenta. Then what is the net charge of glutamate at pH 7? So this is the structure of glutamate. It has got a carboxyl group here, a carboxyl group here, and this is the amino group. Sorry. So these groups, that is the side chain carboxyl, the alpha carboxyl and alpha amino group, they have pKa values as I have written here. So for the side chain it is 4.25, for the alpha carboxyl it is 2.19, and for amino group it is 9.67 pKa. Now if pH is less than pKa, it will be protonated, and if pH is greater than pKa, it will be deprotonated. So they have given the pH value as 7. So 7 is greater. First we will see the charge of the alpha carboxyl group. So 7, it is greater than 2.19. So it will be deprotonated and it will have a negative charge, so -1. Similarly for side chain carboxyl group also, 7 is greater than pKa value and it will also have -1 charge. This is the amino group and the pKa is 9.67. So 7 is less than 9.67, so it will be positive charge, so it will be +1. Now add all the charges, you will get it as -1. So the answer is option 4, -1. Then next one, non-active protein part of an enzyme that requires cofactor for activation is. So it is called as the apo enzyme. Holo enzyme, it is the whole enzyme which is active. It is formed of apoenzyme plus cofactor will give holoenzyme. Apoenzyme, it is the protein portion but it is inactive. And it requires a cofactor for activation. And cofactor, it is a non-protein portion. So the answer here is option 1, apoenzyme. Then next one. Insulin dependent glucose uptake by adipose tissue is the result of. So it is the result of transfer of GLUT transporters from intracellular vesicles to cell membranes. So the transfer of GLUT transporters from intracellular vehicle to cell membrane it depends on the following mechanism of action. First, what will happen is insulin will bind to the receptor and it will trigger a signaling cascade. This signaling cascade it lead to the translocation of GLUT4 from intracellular vesicles to the cell membrane. Then GLUT4 they will facilitate the uptake of glucose into the cells, thereby helping in or increasing glucose entry. Now co-transport of glucose with sodium against concentration gradient as per option 1, it take place in the intestine and kidney, not in adipose tissue. So first option is not correct. Your answer is option 4, that is GLUT transporters from intracellular vesicle to cell membrane. Now next one. Which of the following motor proteins is involved in intracellular transport along microtubules? So it is kinesin and dynein. They are the motor proteins that are involved in intracellular transport along microtubules. They are involved in intracellular transport mechanisms such as vesicle transport, organelle movement, chromosome separation during cell division. Then kinesin it move towards the plus end of the microtubule and dynein move towards the minus end. Then next one. Which of the following organelle is involved in Tay-Sachs disease? Tay-Sachs disease, as you all know, it is a genetic disorder and is caused by the deficiency of the enzyme hexosaminidase A. And this enzyme it is found in the lysosome. So the cellular organelle that is involved in Tay-Sachs disease is lysosome. Answer is option 2. During DNA repair, which of the following enzyme is involved in phosphodiester bond formation? So it is the DNA ligases that are involved in phosphodiester bond formation, not only during repair but also during replication and recombination. Which is not true about endotoxin? So this question was dropped. Which is not true about endotoxin? They have dropped the question. Then next one. Upon re-exposure to a previous pathogen, which immune response will be most likely the fastest one? So they have asked upon re-exposure. So first time there was exposure and so B cell T cells were activated. They get converted into memory B cells and T cells. So upon re-exposure, the memory B cells and T cells will be activated at a faster rate. So innate immunity, it provides immediate defense, but it does not respond. But its response is not specific to the pathogen and doesn't retain memory. Then complement activation, it is a component of innate immunity and it helps eliminate pathogen, but its response is not accelerated by because of memory. Then adaptive immunity and immunological memory, that is correct answer. Adaptive immunity is by T cells and B cells and they retain memory of the pathogen and it will cause a rapid response. Then immunological memory is by memory T cells and B cells and they will retain memory and they cause a rapid response. Killer T cells, they are part of adaptive immunity and rely on immunologic memory. Then next one. What is true about MHC class 2? So MHC class 2, they present extracellular peptides to CD4 plus cells. That is your T helper cells. So the answer here is option 3, whereas MHC class 1, they present peptides from intracellular sources to CD8 plus T cells, that is TC cells. Then next one. Patients with galactosemia are advised not to take dairy products because. Now galactosemia, it is also a genetic disorder and it affects the body's ability to metabolize the galactose. And people with galactosemia, they lack the enzyme galactose 1 phosphate uridyl transferase and this enzyme it is necessary to convert galactose into glucose. Now lactose it is a dairy product, or I can say dairy products contain lactose, and lactose breakdown during digestion will give rise to glucose and galactose. So people with galactosemia they cannot digest galactose, and it will lead to various metabolic disorders or damage to the liver, kidney and other organs. So patients with galactosemia cannot consume dairy products. So the answer here is they cannot metabolize galactose, digestion product of lactose. Answer is option 2. Glycerol kinase is not expressed in. So it is not expressed in adipose tissue. And glycerol kinase it converts glycerol to glycerol 3 phosphate, and it is not expressed in adipose tissue. Then next one. In prolonged starvation, a minimum glucose level is maintained for supplying glucose to brain and RBCs. This is achieved by. So during prolonged starvation, the body's main aim is that it should supply or it should maintain a minimum glucose level for the brain and the RBCs. So gluconeogenesis, that is the process of generation of glucose from non-carbohydrate sources, will maintain the blood glucose level, and this occurs in the liver and the kidneys. So the answer here is option 2. Glycogenolysis in liver, as in option 1, it breaks down glycogen to glucose, but liver glycogen stores are depleted within few days of starvation. So they have asked for prolonged starvation, not few days of starvation. Then third option, glycogenolysis in muscle, it occurs during short-term high-intensity exercise, not during prolonged starvation. Then gluconeogenesis in muscle and liver. Muscle does not contribute to gluconeogenesis. Then next one. Which of the following is a cis-acting element? So an element it is considered cis-acting if it can affect other elements on the same DNA molecule on which it is present. That is called as a cis-acting element. So CAMP response binding protein, these proteins are transacting. So it is not cis-acting. Then operator is cis-acting. It is present on the same DNA molecule as in case of lac operon, in which it is acting. Then repressor is not found attached to the lac operon genes, but it is found near to the lac operon genes, not a part of the operon system of lac operon. So it is not a cis-acting element, it is transacting. Then terminator is also a transacting. Then next one, oligomycin blocks electron transport chain by. Oligomycin is for ATP synthase. So wherever ATP synthesis is prevented, oligomycin is acting. So inhibition of ATP synthesis by preventing influx of proton through ATP synthase. That is the answer. Answer is option 2. Then next one. How many ATP molecules are produced for each molecule of FADH2 oxidized via the respiratory chain? So FADH2, as you all know, it enters the electron transport chain at complex 2. Bypassing complex 1. So it yields only 2 ATP molecules. Answer is option 4. Then next one. In E. coli, PUR repressor, that is purine repressor, regulate the expression of more genes as compared to the lac repressor because. So purine repressor, it regulates expression of gene mainly involved in purine nucleotide biosynthesis, and lac repressor, it regulates gene involved in lactose metabolism. You all know that. Now purine repressor, it does not require a co-repressor. it does require a co-repressor. So option 1 is not correct. The co-repressors for purine repressor are hypoxanthine and guanine. Then purine repressor is expressed at much higher level. That is also not correct. There are more purine operator sequences in the E. coli genome. That would be correct. Purine repressor is expressed at much lower level. That is also not correct. So the answer here is option 3. Then next one. Glomerular filtration is determined by creatinine clearance test and inulin clearance test. Which of the following statement is true about GFR? GFR is glomerular filtration rate. So this filtration rate, it is a measure of the kidney function and it represents the volume of fluid filtered by the glomeruli per unit time. Now both creatinine and inulin test, they are used to estimate GFR. Now inulin, it is a polysaccharide that is freely filtered, neither reabsorbed by the or secreted by the renal kidney tubules. Whereas creatinine clearance test, it has some limitations because it is also freely filtered from the glomeruli and partially secreted by the renal tubules. So this tubular secretion of creatinine, it can lead to overestimation of GFR. So inulin clearance test is the gold standard and is the most accurate than creatinine clearance test. So first option, that is inulin clearance, gives the exact measurement of GFR is correct. Then next one. In hepatocellular disease, both conjugated bilirubin levels are increased. Only unconjugated bilirubin levels are increased. Both conjugated and unconjugated bilirubin levels are increased. Conjugated bilirubin levels are increased. Unconjugated are decreased. So in hepatocellular diseases, that is in hepatitis and cirrhosis, the liver's ability to process and transport bilirubin is impaired. So this leads to the accumulation of both conjugated and unconjugated bilirubin in the blood. So the answer here is option 3, that is both conjugated and unconjugated bilirubin levels are increased. Then next one. AST by ALT ratio is decreased in. So it is decreased in acute hepatocellular damage. Answer is option 2. AST-ALT ratio, it is also known as D-Reitz ratio and it is useful indicator of liver damage. So AST is aspartate transaminase and it is found in liver and muscle cells. Then ALT is alanine transaminase and it is more specific to the liver tissue. So in acute hepatocellular damage that is caused by viral hepatitis, the ALT level rises. ALT, that is alanine transaminase, which is more specific for liver, it rises more than the AST level and this result in decrease in the AST-ALT ratio. So the answer here is acute hepatocellular damage. Now if you take option 1, that is myocardial infarction, that is heart attack, AST is released from the damaged heart muscle, increasing AST-ALT ratio. Then damaged kidney, if you see, neither AST nor ALT is significantly affected. These enzymes are not primarily found in kidney. Then next one. At 50% saturation, each Hb molecule has dash bound. How many molecules of oxygen are bound at 50% saturation of Hb molecule? Now Hb, that Hb is hemoglobin. So hemoglobin, it is a tetramer and it consists of 4 subunits, each capable of binding 1 oxygen molecule. Therefore, single hemoglobin can bind 4 oxygen molecules. Now at 50% saturation, that is half of the sites are bound by oxygen. That is out of 4, 2 are bound by oxygen. So at 50% saturation, each Hb molecule has two oxygen molecules bound. Answer is option 2. Then internal respiration occurs in. So internal respiration. It occurs in tissues throughout the body. Answer is option 4. Which of the following factors affects the affinity of hemoglobin for oxygen? So it is pH, temperature and binding of 2,3-bis phosphoglycerate. pH means low pH decreases the hemoglobin's affinity for oxygen while high pH increases the affinity. Now temperature means increased temperature decreases hemoglobin's affinity for oxygen. 2,3-bis phosphoglycerate means high level of 2,3-bis phosphoglycerate decreases hemoglobin's affinity for oxygen. So the answer here is option 4. Then next one. To solve the mystery of spoilage of wine, Pasteur observed yeast and bacteria in the fermented grape juice, that is wine. Through a series of experiments, he proved yeast and bacteria are required for fermentation. No, only yeast is required for fermentation, not bacteria. Yeast is required for fermentation or alcohol formation and bacteria results in spoilage of wine. That is correct. Then next one. Penicillin, the first found natural antibiotic, acts by. So penicillin, as you all know, it inhibits cell wall biosynthesis. So the answer is option 2. Then next one. Some microbes survive or grow at very high temperature because of heat shock protein synthesis. Yes. Molecular chaperones to prevent or repair protein denaturation. That's also correct. Thermostability of enzymes. Yes, as you can see in the case of Taq polymerase enzyme. So heat shock protein synthesis, molecular chaperones to prevent or repair protein denaturation and thermostability of enzymes. So the answer is option 4. Then next one. Who was the first scientist to prove the germ theory of disease? So it was Robert Koch. Answer is option 1.